And what we found was that there was a 31% risk reduction in the patients having 6 months sustained progression of disability while under treatment with Tolebrutinib compared to placebo.
Brain atrophy may not be an excellent measure of disability progression, particularly with kind of the changing face of MS that we see today.
We seem to have found a therapy that slows the little by little progression that we see in our progressive patients.
But I do wish that we had a little more freedom to be able to use a drug more liberally when we do think that an individual patient in front of us has a disease phenomenon that we think would benefit from a drug like this. Hi, my name is Jiwon Oh, and I'm a neurologist at St. Michael's Hospital at the University of Toronto.
And I'm Bob Fox. I'm a neurologist at the Mellen Center for Multiple Sclerosis at the Cleveland Clinic here in Cleveland, Ohio in the United States.
And I'm Brett Drummond. You're listening to the ECTRIMS podcast, the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. Over the past few years, MS researchers and neurologists have closely followed the development of a new class of therapies called Bruton's tyrosine kinase inhibitors, or BTKis. Multiple clinical trials have been conducted that have provided much food for thought on the potential use of these treatments in MS. Considering this, the announcement that the Tolebrutinib trial results would be presented at ECTRIMS 2024 was met with great excitement. Recently, the full results from these pivotal trials have been published in the New England Journal of Medicine. My guests in this episode, Bob and Jiwon, have led these trials and presented them in the late-breaking session at ECTRIMS 2024. Today we'll be taking a deep dive into those trial results and discussing the next steps for this exciting new therapy. Bob, Jiwon, thanks for joining me.
It's great to be with you.
Nice to be here.
So as a starting point for this, I think it's really important that we go through the trial setup of this because it's going to be important when we analyse the results. So can you both go through the Hercules and Gemini studies in terms of cohort population, length of trial, outcome measures that were used?
Sure, Brett. So I can start by going over the Gemini 1 and 2 clinical trials, which were the two phase 3 clinical trials that looked at the efficacy of Tolebrutinib in people with relapsing MS. So the inclusion criteria of these two clinical trials were pretty typical of a contemporary relapsing MS clinical trial, where it included people between the ages of 18 and 55 who had a diagnosis of relapsing MS, and the maximum EDSS score was 5.5, and they had to have at least some clinical or MRI evidence of relapsing disease in the year or two prior to inclusion. And so in this clinical trial, it enrolled people that met these criteria and looked at the efficacy of Tolebrutinib in comparison to an active comparator. And this active comparator was teriflunomide, which is a pretty common disease-modifying treatment that many clinicians around the world use first line for the treatment of relapsing MS. And so because of these inclusion and exclusion criteria, what we saw with the baseline characteristics of participants in Gemini 1 and 2 were pretty much what we expect in a contemporary relapsing MS clinical trial, where on average, most people were in their later 30s. The vast majority of people had relapsing remitting MS, but there were a very small proportion of people that had active secondary progressive MS. Most people had pretty minimal disability at baseline with a median EDSS score of 2. And actually, around 60% of people were treatment naive at baseline.
Now, the Hercules trial was a trial focused on secondary progressive MS. And the largest unmet need with secondary progressive is patients without relapses. And so this trial focused on just that. In order to get into the trial, patients had to have secondary progressive MS, meaning little by little progression of disability over the previous year or so prior to the trial. Patients also had to have no clinical relapses in the last 2 years of the trial. And indeed, patients were a little bit different than the Gemini trial. The average age at enrollment was the late 40s, about 49 years. About 60% 80% women. The median EDSS score was 6, so about half of patients needed a cane or more for walking. The time since onset of MS was about 17 years. And although the efforts were made to try to minimize the amount of disease activity in that no relapses were allowed, there was about 12-13% of patients who had gadolinium-enhancing lesions at baseline.
So obviously two different setups in terms of the trials, which is, I guess, interesting to give us some comparison. So maybe let's start with the Hercules trial. What were the main findings that came out of that trial?
So the main findings was that Hercules, and we don't have a therapy that has demonstrated efficacy in non-relapsing secondary progressive MS. So the comparator in this trial was to a placebo, to a non-active comparator. And the primary outcome was time to 6-month confirmed disability progression. The disability progression measured in the standard fashion using the expanded disability status scale, and the increase had to be sustained for at least 6 months or more and could not have occurred in the setting of a clinical relapse. And what we found was that there was a 31% risk reduction in the patients having 6 months sustained progression of disability while under treatment with Tolebrutinib compared to placebo. And so this is the first time that we have seen a drug in this patient population slow the little by little progression of disability. We also saw secondary outcomes, the 3-month confirmed disability progression, so a little bit shorter period of requiring the confirmation was also reduced, this time 24%. There was also a reduction in the new enlarging T2 lesions on MRI. Now, this was about 38%, which compared to our standard MS therapies was not as much as we're typically seeing with our new and emerging MS therapies.
Yeah, very exciting in terms of this result. In terms of the Gemini studies, Jiwon, what did we see with those in the relapsing or remitting population?
So in the Gemini studies, because these were 2 phase 3 trials that looked at people with relapsing MS, as expected, the primary endpoint was annualized relapse rate. And when this was evaluated, there really was no difference in annualized relapse rate reduction for Tolebrutinib versus teriflunomide in Gemini 1 or Gemini 2 or in the pooled analysis. So the annualized relapse rate at the end of the study was overall very low and ranged between 0.11 and 0.13 for each study arm that was evaluated. Because the primary endpoint was not met, all of the secondary endpoints, their p-values are thereafter considered nominal. Nonetheless, I think it's really informative to look at these analyses, just because given what Bob just said about the Hercules trial, I think it's really, really interesting. So when confirmed disability progression was evaluated, so 6-month confirmed disability progression, despite the fact that Tolebrutinib did not show a difference in reduction of annualized relapse rate versus teriflunomide, when you looked at 6-month confirmed disability progression, there clearly was a reduction in Tolebrutinib treated patients versus teriflunomide treated patient. And when you looked at the 6-month CDP, that reduction was 29%, and the hazard ratio did not overlap 1. Similarly, when you look at 3-month confirmed disability progression, Tolebrutinib treated patients showed a 27% reduction in comparison to teriflunomide treated patients with a hazard ratio that did not overlap 1. So this is all really interesting because, again, despite the fact that the primary endpoint was not met, so Tolebrutinib and teriflunomide essentially seem to have similar effects on reducing annualized relapse rate. It's really striking that the secondary endpoints of confirmed disability progression, regardless of whether you're looking at 6-month or 3-month CDP, was clearly lower in people treated with Tolebrutinib in comparison to teriflunomide.
Yeah, and I think that's really important to focus on, because obviously when we have the primary outcome measure, we can look at this and just say, well, the trial didn't work. But I think when we delve deeper, there is some indication that this is certainly worth looking more into. One of the results that I'd like you both to talk a little about from these trials was the brain volume loss MRI measures that we looked at, because I thought that was an interesting outcome. So if you could both talk a little bit about what you saw in the trials with that, and whether or not those results surprised you at all.
Yeah, so the brain volume results from Hercules, the secondary progressive trial, found that tolebrutinib did not slow the progression of brain atrophy compared to placebo. Now, the absolute difference, there was a slight slowing, about a 12%, but because of the variability of brain atrophy assessments, was not statistically significant or anywhere close to statistically significant. But it does point us into a general direction that it looked to be supportive without being statistically significant. That was very surprising, because we up to this point had thought that whole brain atrophy was a good biomarker of progression in progressive MS and probably also relapsing MS. And this has caused that to be questioned now, in that we have a drug that slows disability progression, but not brain atrophy. And so I think we have unlearned what we thought we knew, which is that whole brain atrophy was a reasonable biomarker of progression in progressive MS.
And Brett, similarly in Gemini 1 and 2, when whole brain atrophy was evaluated, in Gemini 1, actually tolebrutinib did show a reduction in the rate of brain atrophy in comparison to teriflunomide. But in Gemini 2, there really was no difference, similar to what was observed in Hercules. And so, I think the bottom line is, from these studies, it may be that, obviously it depends on the population you're looking at and at what stage of disease you're looking at brain atrophy. But brain atrophy may not be an excellent measure of disability progression, particularly with kind of the changing face of MS that we see today.
And I think that's a really important outcome, because you both say this is something that certainly we'd been relying on, or certainly thinking would be a really reliable outcome measure in terms of a measure of disability progression. So an important outcome from this trial as well, I think. So thinking about potential mechanisms of how this may be impacting on MS, what can you tell us in terms of what we think tolebrutinib's mechanism of action might be? Certainly with the BTKis, we think one of the biggest things is their ability to really access across the blood-brain barrier and get to sites of importance. I'd be interested to hear both of your thoughts in terms of what you think might be happening there.
So, Brett, as you said, I think we have a lot of information on what BTKis can do. First of all, we know that BTK is a pretty widely expressed enzyme, but we know that this is an enzyme that plays a key role in modulating B cells, as well as innate immune cells, including microglia, which we know plays a key role in driving progressive disease biology. And so knowing that these BTK inhibitors can beneficially modulate two types of cells that we know B cells play a role in both relapsing and progressive disease, and B cells also play a role in progressive disease, but also microglia play a dominant role. So theoretically, there is the idea that if we can get these BTK inhibitors to the right place, it could potentially beneficially modulate processes that are responsible for both relapsing and progressive disease. So the thing about tolebrutinib is there's many different BTK inhibitors being studied, but we have very good evidence that even after a single oral dose of tolebrutinib, it pretty easily gets across the blood-brain barrier. And so there are, really good concentrations that can be measured in the central nervous system, in the cerebral spinal fluid. And really importantly, we also know that these concentrations are at a relevant level to modulate both B cells and microglia within the central nervous system. So, that piece of getting into the central nervous system is one thing, but we also also have to remember that you have to get into the central nervous system at an appropriate dose to target the cells that we want to. So we at least know that not only does tolebrutinib easily get into the CNS, but it gets into the CNS at relevant concentrations to beneficially modulate those B cells and microglia that we were talking about earlier. So given all of this theoretical information, as well as what we see, especially in the Hercules clinical trial, but also with the secondary endpoints of the Gemini 1 and 2 clinical trials, I think we can safely hypothesize that tolebrutinib is likely modulating B cells in the periphery, although, as we already talked about, it probably doesn't have a really highly effective effect on peripherally mediated inflammation, but then it gets into the central nervous system, and at least based on the disability outcomes that we see, it does likely modulate some of those drivers of progressive disease biology that current therapies are not that good at doing. So probably it's microglia and B cells that it's beneficially modulating to result in the disability outcomes that we see in Hercules as well as Gemini 1 and 2.
Now, that's the good news. The bad news is we had thought BTK inhibitors would have a great impact on peripheral B cell function and have a greater benefit on the relapsing pathology that drives relapses and new lesions and MRI in our patients. And it turns out it doesn't really do that very much or very well. It certainly does not equate or come close to what our B cell depleting therapies do when it comes to B cells. So the good news is that it seems to work within the central nervous system, probably on microglia, maybe also on B cells within the CNS. The bad news is it doesn't have as much of an impact on the peripheral B cells and the peripheral immune system. It may explain why another BTK inhibitor, evobrutinib, also didn't work in relapsing MS and did not slow disability progression. And the second part, because evobrutinib does not seem to get into the central nervous system at biologically relevant concentrations. And so explains why that drug's relapsing trial did not have the disability slowing that the Gemini 1, Gemini 2 trials showed, because tolebrutinib can get into the central nervous system.
I think it's important as part of the discussion because it's certainly something that's received a lot of attention throughout all of the different trials that have happened, is a quick discussion around the safety profile of tolebrutinib. I know there's been liver issues that have arisen as part of it. Can you talk about what the trial results for Gemini and Hercules showed in terms of the safety profile?
Maybe I'll go first with Gemini, and this was a signal that was seen across both the Gemini and Hercules clinical trials, but not only with tolebrutinib, but it does seem like with additional BTK inhibitors that are being studied, there clearly is a liver safety signal. When you look at the liver signal in Gemini 1 and 2, keep in mind that because there was the active comparator arm of teriflunomide, if you look at any elevation of liver enzymes, 3 times or more the upper limit of normal, the proportion of people was actually pretty similar in tolebrutinib treated patients versus teriflunomide, around 5 to 6%. But where things differ is when you look at people that had a really high elevation of liver enzymes, 20 times the upper limit of normal, the proportion of individuals treated with tolebrutinib that showed this very high elevation was 0.5%, whereas in people treated with teriflunomide, it was only 0.1%. So the bottom line is it is pretty clear that this liver enzyme issue can be seen. All of these cases, the really high elevations were seen within the first 3 months, and at least in Gemini 1 and 2, once the drug was discontinued, this resolved without any issues for the patient. But very clearly, this is something that we will have to monitor.
Now, this is the good news in that we have a clearly defined period when that very severe elevation in liver enzymes occur, and that's within the first 3 months. So we're hoping that weekly monitoring can identify patients right when they start that uptick in the liver enzymes. And the management is very straightforward. Stop the drug. That's all you have to do. We should monitor and ensure that it comes down later. But there's nothing active that needs to be done. There's not a special antidote that needs to be given within a certain period of time. The other thing we see is a slight increase in the rate of infections in the treated patients compared to each of their respective comparator groups, particularly respiratory infections, more frequent COVID-19 infections in those treated with Tolebrutinib compared to placebo in the Hercules trial, as well as upper respiratory infections. So perhaps a slight signal in the respiratory infections.
Look, I think these are all really exciting results, really important results, obviously, especially in the non-relapsing SPMS population, as you said at the very start. This is a huge unmet need. To finish the episode, what do you see as the next steps here for this treatment, both in the secondary progressive MS population and also as a potential use in the relapsing population?
So we seem to have found a therapy that slows the little by little progression that we see in our progressive patients. And as the Gemini 1 and 2 trials also showed, they are seen in our relapsing patients. So they probably have that progression independent of relapse activity, PIRA, in ways that we can't easily see and easily appreciate. So this emphasizes the overlap of progressive MS and relapsing MS and how they occur simultaneously. And there probably is progressive MS at the very, very early inklings of the relapsing, or at least early on in the relapsing remitting disease course. And so figuring out when to use a therapy like this is one of the challenges. Now, we were a little disappointed about the rather modest relapsing therapy effect. So the equal relapse rate to teriflunomide, only 38% reduction in T2 lesions compared to placebo. So it's a very modest anti-inflammatory when it comes to the relapsing component. So in patients who are older, who are in the mid-50s and older, who probably have a lot less relapsing pathology, using this drug is something that could well be considered and discussed with patients. In younger patients, it's a little bit of a struggle. In patients who are in their 30s or 40s, having them stop their current disease-modifying therapy to use this for progression, knowing that they probably won't be as well treated of their relapsing pathology as their current highly effective therapies, that's a struggle in an area that we don't really know how to take this drug at this point.
So I agree with everything that Bob said, and just wanted to add, I think as clinicians, we would like to be able to use a drug like this in anybody who has non-relapsing progressive disease. And this is regardless of what the categorization of that patient is. Because as Bob said, very clearly we do see evidence of progression that is independent of any relapsing disease activity, even in early relapsing MS. This is what not just the Gemini 1 and 2 clinical trials have shown us, but even, the first description of PIRA, that was a post-hoc analysis of the ocrelizumab phase 3 clinical trials. But how the regulators will approve this drug is probably what will ultimately determine how we can use it in clinical practice. And I have to say, as a clinician, it's frustrating just because, we do have certain relapsing MS patients that we currently label as relapsing MS patients who clearly have evidence of non-relapsing progressive disease. And so in somebody who we think is suitable, it would be useful to be able to access a drug like this. It's just that based on the way drugs are approved, at least up until now, it's always approved for a particular subtype or category of MS. And so I guess we'll see, because we don't yet have regulatory approval, but I tend to be an optimist. But when it comes to the regulators, I think almost always things will happen the way they have happened up until now. And so that will ultimately determine how we use it. But I do wish that we had a little more freedom to be able to use a drug more liberally when we do think that an individual patient in front of us has a disease phenomenon that we think would benefit from a drug like this.
In addition, there are several additional BTK inhibitors that are in trials in both relapsing and progressive forms. And so we eagerly await those trial results to understand the more breadth of activity of these other BTK inhibitors across the spectrum of MS.
Certainly an interesting space, and we'll be eagerly awaiting those trial results and also the regulatory approval process, which is already underway with the FDA at the moment. So thank you both for your time, sharing your insights from these trials today and all your hard work in leading both of these two really pivotal trials.
Thank you for having us.
Great. It's wonderful to be with you. Yeah.
Thanks everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode and co-founder of MStranslate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with two key partners, MStranslate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The 2024-25 ECTRIMS podcast series is supported by educational grants from educational grant providers, including Alexian AstraZeneca Rare Disease. Educational grant providers have no input in the podcast content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on X, formerly Twitter, LinkedIn, Facebook and YouTube.